Welcome to our video on acids and bases, pH and titration. In this video, we will explore the fascinating world of acids and bases, their properties, and how they interact with each other. We will also discuss the concept of pH and titration, which are essential tools in chemistry. Acids and bases are two types of chemical substances that have opposite properties. Acids are substances that can donate hydrogen ions H+ to a solution, while bases are substances that can accept hydrogen ions. The pH scale is a measure of the acidity or basicity of a solution, ranging from 0 to 14. A pH of 7 is considered neutral, while values below 7 are acidic and values above 7 are basic. Acids and bases are present in our daily lives, from the food we eat to the products we use. For example, citrus fruits such as lemons and oranges are acidic, while baking soda and soap are basic. Understanding the properties of acids and bases is crucial in many fields, including medicine, agriculture, and environmental science. In the next segment, we will discuss the properties of acids and bases in more detail. Acids and bases have distinct properties that differentiate them from each other. Let's start with acids. Acids have a sour taste and can corrode metals. They turn blue litmus paper red and have a pH value less than 7. Acids can react with bases to form salts and water, a process known as neutralization. Some common examples of acids include hydrochloric acid HCl, sulfuric acid H2SO, and acetic acid CH3Cu. On the other hand, bases have a bitter taste and feel slippery to the touch. They turn red litmus paper blue and have a pH value greater than 7. Bases can react with acids to form salts and water. Some common examples of bases include sodium hydroxide NaOH, potassium hydroxide KOH, and calcium hydroxide KOH2. Acids and bases can also be classified as strong or weak. Strong acids and bases completely dissociate in water, while weak acids and bases only partially dissociate. The strength of an acid or base is determined by its dissociation constant, which is a measure of how much it dissociates in water. In the next segment, we will discuss the concept of pH and how it relates to acids and bases. pH is a measure of the acidity or basicity of a solution. It is defined as the negative logarithm of the hydrogen ion concentration in moles per liter pH equals log H+. A solution with a pH of 7 is considered neutral, while solutions with a pH less than 7 are acidic and solutions with a pH greater than 7 are basic. The pH scale is logarithmic, which means that a change of 1 pH unit represents a tenfold change in the hydrogen ion concentration. For example, a solution with a pH of 3 is 10 times more acidic than a solution with a pH of 4. Acids and bases can affect the pH of a solution. When an acid is added to water, it donates hydrogen ions to the solution, increasing the concentration of H+ ions and decreasing the pH. Conversely, when a base is added to water, it accepts hydrogen ions, decreasing the concentration of H+ ions and increasing the pH. The pH of a solution can also affect the properties of acids and bases. For example, a strong acid in a solution with a pH of 1 will be more acidic than the same acid in a solution with a pH of 3. Similarly, a weak base in a solution with a pH of 9 will be less basic than the same base in a solution with a pH of 11. In the next segment, we will discuss the concept of titration and how it is used to determine the concentration of acids and bases. Titration is a technique used to determine the concentration of an acid or base in a solution. It involves adding a solution of known concentration the titrant to a solution of unknown concentration the analyte until the reaction between the two is complete. The point at which the reaction is complete is called the equivalence point. Titration can be used to determine the concentration of an acid or base in a solution, as well as the purity of a substance. It is commonly used in the pharmaceutical industry to determine the concentration of active ingredients in drugs and in the food industry to determine the acidity of products such as wine and cheese. The most common type of titration is acid-base titration, which involves the reaction between an acid and a base. The titrant is usually a strong acid or base, such as hydrochloric acid or sodium hydroxide, while the analyte is the acid or base being tested. During the titration, an indicator is used to signal the equivalence point. An indicator is a substance that changes color when the pH of the solution changes. For example, phenolphthalein is a commonly used indicator in acid-base titrations. It is colorless in acidic solutions and pink in basic solutions. The concentration of the analyte can be calculated using the volume and concentration of the titrant and the stoichiometry of the reaction. The stoichiometry is the ratio of the number of moles of the titrant to the number of moles of the analyte. In the final segment, we will summarize the key points of this video and provide some additional resources for further learning. In this video, we have explored the properties of acids and bases, the concept of pH, and the technique of titration. We have learned that acids have a sour taste, turn blue litmus paper red, and have a pH less than 7, while bases have a bitter taste, turn red litmus paper blue, and have a pH greater than 7. We have also learned that pH is a measure of the acidity or basicity of a solution, and that titration is a technique used to determine the concentration of an acid or base in a solution. Acids and bases are present in many aspects of our daily lives, from the food we eat to the products we use. Understanding their properties and interactions is crucial in many fields, including medicine, agriculture, and environmental science. If you would like to learn more about acids and bases, pH, and titration, there are many resources available online. Some useful websites include Khan Academy, ChemGuide, and the Royal Society of Chemistry. Thank you for watching this video on acids and bases, pH and titration. We hope you have found it informative and helpful.